Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi, everyone. I'm excited to be here today with Professor Tim Spector, who's Professor of Genetic Epidemiology at King's College London and co-founder of Zoe, which you may have heard of if you're interested in personalized nutrition. I heard of them when my wife had me eat a blue muffin that we can talk about a little bit later on in the show. But Tim is also famous for some of his pioneering twins genetics research in what I would call maybe a previous chapter of your career. So we're going to cover what we learned in the twins genetics era, Tim's perspective on the field today, and where it's going. So welcome and thanks for taking the time to be with me.
Great to be here.
I'd love to start with the first chapter of your career, or if there's a chapter before you really pioneered some of the earliest twin studies, then let's start there. But maybe you could talk a little bit about the major programs that you led and what we learned in that era of genetics research that still is pretty fundamental to what we understand about many common diseases today.
Well, before my genetics phase of life, I was a rheumatology MD. And so I was a consultant rheumatologist, specializing in autoimmune diseases, osteoporosis, arthritis, etc. And was doing research epidemiology in that field and took 3 years out to do epidemiology. And that got me interested in population type research. And I'd finished my thesis and then was thinking of a new area to go into, so I wasn't competing with my supervisor who was in the same field and had got more money than I had. So I wanted to do something else, and I decided after a few months of going around asking people what to do a twin study, and that was in 1992, and there was no twin study in the UK at the time. The previous ones had really folded for political reasons because they were told that in sort of socialist Britain, talking about IQ being heritable and things like this were just too hot topics. That was the time of academics being cancelled, back in the 70s.
Interesting.
It's quite interesting. So they left for more right-wing countries, and there was nobody doing twin research in the UK. So that's when I started and built that up, initially to look at rheumatological diseases, but then realized you had the same method. You might as well study every disease if you were bringing people up for a clinical exam. So that is how it started in those days, we didn't really have good markers for genes. And so it was all the heritability studies, nature v nurture.
Yep.
And no one had really done this for large numbers of diseases. So you've got to remember back then, people just thought rare diseases were heritable and all the others were boring and were just due to aging. So that we spent the first 10 years busting myths about these diseases. So we found that back pain was more heritable than breast cancer.
Huh.
We found that,
Really?
3 times more heritable. So a lot of these common conditions, which people thought were wear and tear, ended up being really important. Now that we published New England Journal of Medicine about cataracts being heritable, and previously that was just old age. So a lot of these old age things were told totally inevitable and just random chance turned out to have some genetic basis about why people got them. So that was a lot of the work that we did. And we went from the physical diseases, to metabolic diabetes, to even things in personality, behavior, um, even things like, politics, right-wing views, being heritable, um, and personality traits and sexuality, all kinds of anything that hadn't been done before, I was interested in looking at. So it broadened my mind really, and moved me away from my small speciality into this big wide area. And then we got into gene discovery. Went through the linkage era in genetics, which was actually a bit of a dark period because for 10 years people were publishing essentially nonsense,
Right.
And candidate genes. And it wasn't until we got to the big GWAS studies, of the early 2000s, about 2007, that then we started getting really precise answers. And then it became clear slowly that there wasn't one gene for any disease, there were 1,000 genes for every disease. And so the old ideas you could predict common diseases from genes went out the window, but you could certainly learn a bit about mechanisms of disease, which we hadn't thought about. So as we got these results, it changed our view about what we could do. But as someone who wanted to, I don't know, have a practical use out of what I was doing, I got a bit, although it was really good for my CV and I got lots of papers in Nature Genetics, it wasn't as satisfying as I thought it would be. So I started looking for other things within the twins that would be interesting. Spent a few years looking at epigenetics, the differences between identical twins, and but the technology really wasn't great there. And so it was in about 2011 I did our first study on twins and microbiome, and then that was the first thing that we saw that was different in identical twins that might explain why we're all more different than our genes in how we respond to our environment. So it's been a bit of a journey. And then from the microbiome, I got into nutrition and realized that if the discovery of the microbiome suddenly made nutrition one of the most exciting areas of science rather than the most boring.
Right. Yeah, we're going to dive deep into that area. Before we do, I have a question that I thought you'd be really well placed to answer, which is around the concept of missing heritability. Having spent years doing some of the seminal studies in genetic heritability based on twin studies, there's a concept that we still talk about at major genetics conferences today, which is the gap between the twin studies and GWAS studies. I want to get your perspective on what you think are the big drivers of that. Is it a settled question at the moment, or is it still an open question of why we see such a big gap between GWAS studies and some of the studies you led?
I think it depends who you ask about whether it's a settled question. So if you ask the twin people, they'll say the twin studies are correct and that there's something that you're missing in the GWAS that you're just not picking up the full extent of the genetic inheritance. And then you ask the GWAS people, they'll say twin studies are overestimating the real effects. GWAS is the unbiased effect.
Yeah. What do you think?
Probably somewhere in the middle. I've been party to both camps,
Yeah.
having done both studies. I think what trouble is, nearly everything's heritable, including our environment.
Right.
So this is kind of odd concept, but nearly everything we studied, like, do you like participating in sports? Do you smoke? Do you drink? Do you like unhealthy foods? They all have some heritable component. So probably in twin studies, we are bringing that side of things into the equation that the standard GWAS is not getting. But plus, there's a lot, there's still large bits of dark matter of the genome that we don't understand. that are probably shared by twins that are not being picked up or interacting in certain ways. Um, but I think, certainly as I've gone on, the twin studies do seem to be slightly inflated, is my view from afar. But I think GWAS studies alone are probably underestimates. So we're probably sitting somewhere in the middle with these
Right.
different concepts, the heritability of the environment, that we sort of select environments based on some preferences we have genetically, which goes into that
Mm.
model, and this sort of interesting dark matter that we still don't understand, and the whole, and also, this the epigenetic control of whether those genes are actually switched on or off.
Yeah.
So, we'll find that in 20 years' time, we'll laugh and think, well, we really didn't understand much about that, did we? But
Yeah.
it is interesting now how this heritability question sort of does, yeah, been in it sort of three decades, how it tends to ebb and flow.
Yeah.
And definitely it's looking like, oh, nothing's very heritable these days. It's all environmental now,
Right.
which is quite funny because I've seen it, you know, from the days in the 1970s when we didn't really know anything about genes, going full circle.
Do you think it seems like most of the investment in the last 10 years has gone into mega GWAS studies as opposed to like a mega twin registry, for example. And I'm curious to get your take on if there was an alternate universe where we had mega twin registries. I think you had around 15,000 in Twins UK. I think there's a Swedish program that has around 200,000, but suppose there was an international registry that had 500,000 on the scale of the UK Biobank. Do you think we would learn something profound there, or were you hitting marginal benefits from the tens of thousands or low hundreds of thousands with that approach?
Well, the problem was that ours was the biggest registry that had clinical visits on everybody, that had blood draws on everybody, that had body scans on everybody. Things like the Swedish twin registry or the Californian twin registries were often questionnaire database only,
Right.
or record linkage only. So, I mean, the thing that I would have loved to have had would have been more cases of, say, discordant identical twins.
Mm.
If you had thousands of discordant identical twins for multiple conditions, I think that would have been an amazing data set to have where you could look at multiple omics across the board, really pick out, that would sort of settle a lot of epigenetic questions that you can't really settle in these large, you know, unrelated cohort studies. And you could have done that as a biobank. That's what I would have put my money on.
Yeah, interesting.
Because you've got 100% genetic concordance, and then you're looking at the differences. And in a way, I think the differences are the most interesting,
Yeah.
as opposed to the similarities.
Yeah, that's interesting. Is anybody doing that today that you know of?
No. Well, in small numbers, yes, but we've been having forced to work in like 10 or 20 pairs, and it's just not enough.
Yeah. Yeah.
So
Interesting.
we're always getting borderline significant results. But it's always fascinating, even at the anecdotal level.
Yeah.
And I wrote a whole book really on just anecdotes of, it was got identically different, about looking at identical twins where one had one conditional personality and the other different, and everything from homosexuality differences, or religious differences, or why one was suicidal and the other one wasn't. And I think, we can learn a lot about human diseases from that model. And
Yeah.
it would have really been worth investing in.
Yeah. You're isolating such a major variable. On the story you told earlier about actually seeing the microbiome data for the first time, and that it sounds like that led you into what has been a major chapter in your career. Maybe you could talk a little bit more about what you saw there and what got you so excited at the time.
Yes, I got into the microbiome in 2009, I think it was. I was at um, the um, the annual Cold Spring Harbor genetics meeting, and it was a
Been there a
guy.
few times. Yeah.
Yeah, great meeting, terrible accommodation.
Yeah, I was gonna say it's something special. The food's not great either, but that's okay.
But yeah, it's the meeting to get up to speed on. There was a guy called Marty Blazer giving a talk on Helicobacter, and this was the bug that is the cause for duodenal ulcers that totally changed the whole world of our concept of what an ulcer was.
Mm,
And people have then been trying to eradicate it and saying actually there's a downside to eradicating it. You might get some cancers elsewhere and some other problems elsewhere. And that was the first time really I'd heard of the microbiome. So it took me a couple of years to get connections together, then work with a team at Cornell. And we managed to do a twin study, about 400 twins, looked at their microbiome. It's very early days, of course, using the 16S sequencing method, just a single gene, but that told me enough that there was hardly any difference between identical and non-identical twin similarities in the microbiome. And that to me was a bit of a aha moment.
mm.
Um, really low heritability and in general, much more variation between people than I'd seen in anything else. And that really surprised me because I'd been looking with every metabolite, every chemical blood test, urine test, whatever, it all came back
Interesting.
strongly heritable. So that really changed my mind. I said, well, that could explain the differences between identical twins that we haven't been able to explain with epigenetics or metabolomics or proteomics or gene expression or whatever thrown at it. This was suddenly the big difference, and it just went from there. So obviously it was hard to get funding in the UK, so a lot of it, the early work was done with US collaborators, and we just did more and more microbiome testing in the twins, and eventually, that led to me co-founding a company 8 years ago to do this on a big, massive scale. And now we've got, 250,000 people who've been tested, and we've worked up to, obviously full shotgun sequencing rather than the 16S. And yeah, I haven't looked back really. It's been, great fun using genetic methods to look at the gut microbiome, which is even more complicated than the human microbiome.
Yeah, you've done basic research, you've done translational research, randomized clinical trials. You also have a major consumer group that are really engaged, and so you've been able, I think, to look at this problem from a few different angles. If you were to summarize today the big areas where you'd say it's really clear that the microbiome has a significant in terms of statistics, but also impact on health. What are the big areas where you say it's pretty clear that we should be paying attention to the microbiome here? And then after that, I want to talk about the areas where maybe there's still question marks. We need more data, or we need different tools to understand what exactly is going on.
Well, I'm, I'm pretty clear it's important in all aspects of human health. The question is, how much has the science kept up? Can sort of in an academic way,
Yeah.
I can sort of say it's been proven, and also working out relative to what, So, um, but in general, I think what's coming out is the importance of two real systems in the body proving to be important: the microbiome and the immune system. And the two are so closely linked now that you really can't separate them. Most of the immune cells are in the gut. They're messaging between each other. There's a two-way system. And if you see that the microbiome is integral to the immune system, then clearly, every source of inflammation, every source of accelerated aging, anti-cancer, and now we know that there's a link between the brain and the immune system, mental health disorders. There really isn't many areas of functioning that don't involve this immune signaling from our gut, and it sort of makes sense really because every animal has a microbiome and a gut, and their immune cells are all around there, their nerve cells are around there, and it's got to be for a reason that they were absolutely core to this central signaling mechanism of the body. And we have, unlike with our genes, we have a clear way of regulating this through what and how we eat. So this is why it is so exciting, and why we can see such dramatic changes in our clinical trials and things, within a couple of weeks, to body functions that 10 years ago you'd say, well, that would be crazy to think that you can change your mood or your energy in a few days, just by changing your gut microbes. But yeah, you can. So have we proven it all? No. But we've got some nice animal models that you can test theories in. It doesn't mean it happens to the same degree. I learned this in epigenetics. There's some huge things that happen in mice that don't happen in humans. But what, but we can clearly demonstrate these mechanisms and the direction of the mechanisms in mice, which you can't always do in the humans. So we've got that advantage, and it's just it gives you a real buzz because you can give people practical advice that can really change their bodies very fast, and that's why it's so nice to do clinical trials. Which very hard to do that in genetics, as I was struggling with. But yeah, so, and I think it is changing all the time, and it lends itself to personalization as well. We haven't really talked about that, but obviously I went through the era of personalized genomics, which was supposed to herald all these targeted drugs and things, and apart from a few rare examples, never really took off. Whereas I think personalized nutrition and personalized microbiome, orders of magnitude different, and has much more potential.
To get to the point where you would feel like, yeah, we actually have proven that link. What's missing there? Is there level of resolution that we need from new technologies? Is it better linking the microbiome data to phenotyping differently? Is it simply time to amass bigger data sets that can prove that link? Because I think the mechanism you lay out is clear, right? You have this whole essentially other organ in your body that's dumping out chemicals. Your immune system is picking it up, and your immune system affects absolutely everything. So if that's going haywire, you can understand all kinds of downstream problems. And you also have the gut brain axis that you mentioned. One of the things that has always struck me as challenging about the microbiome is, unlike genetics, it's shifting all the time. So you need serial measurements. You need to figure out how to isolate cause and effect. So I'm curious to get your sense on the technology landscape and whether are there breakthroughs that we're missing that we need to really crack the code, or is it an application of what we have today?
Well, I think we're doing microbiome research at scale. It's similar to the GWAS era. So having gone through linkage studies, candidate gene studies, the 50K chip, which failed, and then finally getting to the point where you had the 300,000 marker chip done in thousands of people. That was the point at which we started to see consistent genetic results.
Mm.
Now we've moved from this era of sort of candidate gene studies with 16S in the microbiome to we're now publishing papers with 30,000 individuals. We've got a database of over 250,000. We can see these clear patterns between microbes, health, microbes, and nutrition. We discovered a microbe that only eats coffee,
Huh.
and it
I've
was
got a
the
lot of those,
strongest.
probably.
Yeah, it was the strongest association. So it's about this. It's probably a unique one that it was the easiest one to spot. It was like this outlying gene, I don't know,
Right.
it was like the ApoE4 of,
Yeah.
or the NOD3 of the GWAS and the area, Larsenobacter, it's called, after Dr. Lawson, and it only eats coffee and only high in coffee drinkers, and it's in tiny amounts in non-coffee drinkers. It's so fussy. So we know we can start to pick these off now. We've got the easy ones, and as the numbers get bigger, we can find the ones for nuts and seeds. We can find the one for broccoli. We can do all this individualized stuff and really start to understand what's going on at a cross-sectional level. Then you need longitudinal studies to say, well, if we give people nuts and seeds, which microbes go up? Do the good gut microbes go up or the bad microbes? If you give them a burger, what will happen?
Yeah.
If you give them antibiotics, which way does it go? So I think scale is huge, is our way around some of the technical problems of accessing the body. So luckily we have a stool sample which you can measure once a day, but you can't measure it every hour.
Yeah.
You can't go in and biopsy normal humans every day. You can do it every now and again. We do know the stool is a reasonable replicate of what is going on in the microbiome. We actually did 200 colonoscopies in twins and showed this.
Huh.
But we can't get anywhere near the small intestine, where there's probably all kinds of interesting immune interactions going on. So we have to sort of guess from mice what's going on there. So there's lots of bits. They're going to be really hard to pin down exactly what's happening inside us, that we have to extrapolate from the big data. But there are tools that are improving. There are these capsules you can swallow that take biopsies on their way down through your gut, called smart capsules.
I was going to ask about that. That exists. That's amazing.
They exist. They used to cost about $5,000 a go, so it was an expensive tablet, but they're getting cheaper. And so I think in a few years they'll be more routine. So you'll be able to get hundreds or thousands of people to do this and get some small intestine biopsy samples that way, which would be really cool.
Wow.
You can measure metabolites in the stool now quite reliably. That needs to be cheaper, but you can do that. And so you can start to compare the chemical products of the microbes. And the other advances, of course, we start to get to the function of the gut microbiome, because it's probably not so much the individual microbial species that are important. It's what chemicals they produce.
Right.
And they're obviously interacting together. It's extremely complex. We're going to need AI and all kinds of other clever stuff to sort that out. But if we can start to classify them by function, then we can start to manipulate it in a much cleverer way than we have before. So I think a lot of the tools are there. I mean, there have been some setbacks. Fecal transplant ended up being much less useful than we thought it was going to be.
Yeah, why is that? I've always been curious about that.
Well, we don't know why it hasn't been successful, other than possibly we're all so different that it's a bit like, we've got so many different blood, equivalent of having so many blood groups that a transfusion from a random person isn't going to work in you.
Right.
That it just doesn't fit into that environment. So until we start personalizing them in a way, the chances of it working are quite small. I mean, it still works for recurrent C. diff infection. It's the treatment of choice.
Mm.
It works to some extent in ulcerative colitis in 1 in 5 people, but the rest of it has been quite disappointing.
Right.
But that was obviously a very crude technique, developed in the 1950s that hasn't really evolved. So, but I think all these other computation advances, in 2 years we like doubled the number of species we can find genetically in the gut. So most of them now are really interesting ones we're finding don't even have a name. So they just got numbers.
Wow.
No one knows about them. So, there's so much out there, but it's coming at us at such a rate. It's very hard to try and work out what the key areas are we should be focusing on. But undoubtedly, it needs to be focused, not make the same mistakes of genetics of just going after something academically interesting.
Yeah.
But it should be geared at, how can we improve people's health? What are the key microbes that we need to improve that are either signs of good health or that actually are doing things functionally that are going to prevent illness?
Is most of the signal in this data coming from like the macro movements of the ecosystem in the sense that you've got big populations of, and I'm oversimplifying here, obviously good and bad bacteria that are pumping out good and bad chemicals? Or is it like there are really rare and niche bacteria that show up in certain circumstances and wreak havoc? I'm sure there's a mix of both, but is there kind of like a common disease, common ecosystem paradigm where for most of the common diseases we see, it's actually like you don't need a microscope to look in and understand that it's some super rare community driving it, but it's more obvious? Or is it that there's actually lots of individually relatively rare but damaging um, bacterial species that are popping up as you look in different diseases?
I think it's more that the common diseases, it's the ratio of the good to bad that seems
Yeah.
to be predominant. Um, I think there are exceptions to this, but they're generally rare. And that this increasing idea that if you've got enough good bacteria in there, they're they're occupying the space, and so they're not leaving any leftover food or nutrition for bad guys to come and multiply.
Right.
And when you lose that environment, then you might get rare or opportunistic microbes coming in. But the key seems to be keeping that core of good microbes in your gut for as long as possible, and they dominate it. And it seems to be, so our researchers, I was originally focused much more on the bad bugs, thinking you've got to get rid of those guys,
Yeah.
the pro-inflammatory ones. But now it's actually much more about how do you keep the good guys
Happy.
healthy and give
Mm.
them the right foods, and they'll do the job for you. So that seems to be true. I mean, there's some interesting stuff in cancer about how um, you might get some unusual bugs uh, cropping up uh, years before with cancer.
Oh,
But,
interesting.
but that might be because the cancer itself is, say, colon cancer is producing a strange environment.
Right.
And so there are like a signal of that really early environment where perhaps the pH has changed or
Yeah.
something unusual is going on. Um, so that, yeah, so that seems to be the way it's going. So I don't think we're going to be looking at, this one microbe predicts this disease. It's more these large patterns.
Yeah.
And we need to be focusing on what those good guys are, how to feed them well, and how, that keeps a really stable environment. So even if you have an infection or, when you go off to to Asia or something and get Asian tummy infections, you bounce right back. You're not, you don't go into some downward spiral. I think it's keeping that stability that's important, which means, eating the right food, not having those antibiotics, um,
Yeah.
and doing all the things to maintain it.
That the cancer point is an interesting one because to your point, it may not be mechanistically interesting in the sense that this bacteria is causing the cancer, but it still can be really interesting from a screening perspective or public health perspective, right? If you have a good biomarker years earlier, to your point about there's applications here that could be really powerful on a consumer or public health basis
And
that
they're
may not.
often seen years before. So
Yeah.
there's this, this this particular Fusobacter, you can see it 10 years before a cancer.
Wow.
And so, looking like tenfold increased risk of colon cancer. So, you know,
Wow.
I think in the future, these will be really useful to be picking up on stool testing to say you need a colonoscopy, quickly,
Right.
because there's something strange going on in your gut.
Yeah, and is there how much work has been done into that? Is it that these are, could they be individuals who are genetically predisposed and you're picking that up, or is it an indicator of a environmental issue or lifestyle that is creating that environment that's going to persist for years and the cancer develops? Do you have a sense of which of those it might be?
No, I don't think there's enough data on that really. They haven't done big
Yeah.
enough studies on it. I mean, we're we're collaborators on a big UK US study on studying bowel cancer in the under 50s to try and get why those rates are
Yeah.
going sort of doubling every 10 years at the moment, and
Wow.
it could be something really weird going on with the environment as well. Could be something as like microplastics or
Yeah.
some other environmental toxins that are create just changing the environment enough so that these pro-cancerogenic um, microbes or producing chemicals that then make it more likely that cancer can grow, or they confuse the immune system. We don't know
Yeah.
quite what it is, but these are really exciting cutting edge applications. Um, but we know that microbes are crucial in how we metabolize all our medicines and pharmaceuticals as well as foods. So if everything's at the chemical level, so
Yeah.
maybe why far more that we used to think genetics were the reason that drugs failed, but it's looking like the microbes are probably much more important,
Interesting.
and getting side effects or inactivating them. Particularly good examples are like taking Tylenol. Many people don't respond to Tylenol because their microbes are deactivating it, um, and only about a third of people respond well to antidepressants. And again, our microbes can actually deactivate the antidepressants themselves,
Wow.
and many other drugs, are implicated. And of course, we know that if you're taking immunotherapy, the state of your gut microbes are probably the most important factor determining your survival. We did a study on melanoma,
Is
a
that
couple of
right?
hundred end-stage melanoma cases, and yeah, when you start the immunotherapy, if your microbiome is not healthy, you won't get that same immune protection from the drug. And so, really, we should be building up the gut health before you go on these drugs. And just unfortunately, the medical profession just hasn't caught up on all
Wow.
the latest science.
What do you think it's going to take to accelerate that translation into the into the healthcare system? Because you named a couple of really good examples here that I think are big opportunities.
Oh, it's hard. I mean, the problem is partly that there's no speciality that's said that we want to take on the
Mm.
microbiome. Um, you know, Microbiologists don't want to know because they only like bad bugs.
Right.
Um, Gastroenterologists don't seem to want it
Mm.
either. Um, Clinical genetics, they're not, although it's a genetic diagnosis, they don't seem to be interested in this either. So
Mm.
it's suffering from a lack of a home,
Yeah.
so no one's championing it. Um, it's also, but if a healthcare system wanted to do microbiome testing, you know, you could get full sequence for less than $50 easily at volume.
Yeah.
And you could screen this. So I think it'd be much more effective, for example, than doing, um, you know, blood testing for colon cancer. You could just simply, change it in that way. But, you know, at the moment, there haven't been enough really good screening, hasn't been put into screening programs properly to test it. Because undoubtedly, when you put it in, the theory is never quite as good as the practice in all
Right.
screening systems. We know that from genetics as well. We always overestimate the benefits. But I think it is now time to really, do a big project and keep the samples and see what happens. But
Yeah.
it's been left out of all the big studies, all the big biobank studies. You
Right.
know,
Why is that? Why do you think
again, you know, nobody championing it. People think it's a fad. Geneticists generally have been against it. They haven't liked it, seen it as competition for funds, maybe.
Mm.
There's always sort of this rivalry.
Yeah.
Anything new, oh, I don't, if that's new, they'll give all the money there. They won't give it to us. Um, yeah, it's hard to explain really when that you see so much good science coming out of it, and you
We're
realize
seeing
it.
them we're seeing at least the UK Biobank, for example, investing pretty heavily in proteomics. I'm just wondering why this would be different.
Well, you have to ask those guys. I mean,
I'll get them on
I
and see. Yeah.
I never got any money out of the UK, for microbiome research. It was the EU and the US where I got my funds. I think they're very old-fashioned, and you know, if there's no history of people doing it there, they feel uh, they still think it's a fad that's going to go away, and so they can understand proteins.
Yeah.
Uh,
What about what we've learned about ultra processed foods, microplastics, those sorts of things? What do we know? What do we not know when it comes to those categories?
we know very little about microplastics and the microbiome. I mean, there's been a few rodent studies and things, a handful of tiny clinical studies. Nothing I would say is reliable. There's reason to suspect they interact with our gut microbiome, um, with I think merits further work, and that's one of the things we're doing in the early colon cancer study is looking at that relationship. I'd love to see some more work there, but we're all we're all full of microplastics, so it's kind of hard to say what are we going to do about it.
Yeah.
Um, I know I tested myself recently, and I've got high levels, but what do I do?
Yeah.
Stop
Where do you get
breathing.
tested for that? Are there companies
Um,
that do it?
yes, there's a company in the Netherlands that I mean, I did it in London. There was a clinic in London, a private clinic was offering some free testing for publicity, so I did it.
Cool.
Got the bad news that I had high levels, but
Right.
you know, um, stop drinking plastic. Uh, but the other ultra processed food, that there's much more data on that, and uh, clear evidence from human and mouse studies that a lot of the components of ultra processed food are harmful for our gut microbiome. So we're talking the additives like the emulsifiers, which can affect the microbes and the gut lining. You've got the artificial sweeteners, which nearly all interact with the gut microbiome in some respects, some worse than others. So particularly aspartame, saccharin, ACE-K, seem to have really quite marked effects on the microbiome. And in certainly mouse studies can produce diabetes type signals. So where people are taking it to stop the risk of diabetes, and these actual chemicals are increasing the risk. And I think all these chemicals that aren't, a lot of those come from the petroleum industry. So our gut microbes have not evolved to be eating them.
Right.
Send off weird signals that we don't really know what they do. So I think this is another reason that it's in a way a mechanism of another mechanism why ultra-processed food is bad for us. It's not just the calories and the overeating, it's the fact they are messing up our microbes, which are messing up our immune system, which is then giving us low-level inflammation. And I think that's one of the mechanisms by which they're harming us. So I think really, really important. And if we didn't know about the gut microbiome, I don't think we'd be as anti-UPFs as we are now.
Mm. As we wrap up here, I just want to ask you about one more thing that's coming out soon. I understand you've got a new microbiome score that you've developed. How are we communicating and scoring the health of the microbiome today, and what about that is are you looking to change and need to change?
Well, for the last 20 years, we've been using something called diversity. So this alpha diversity score that is just saying, well, how many, which is in a way of saying, well, how many different species have you got within your own gut? And so what is the range of species? And it was a reasonable way of doing this and stood the test of time. People who had a small range of species ended up with many more chronic diseases, and people with large ones were generally protected. And there was quite a good correlation with this that really worked pretty well across populations. Didn't matter which ones they were, and therefore it was a really good global method we had. But it didn't work very well cross-sectionally. It didn't work in trials longitudinally. Often you'd give people artificial fiber supplements, and the diversity score would drop or didn't improve. So many trials failed because it didn't get that change. Because sometimes you might be just improving the good ones, but the bad ones, would drop, and so you wouldn't actually get a change in diversity. Right. So we've come up with a new score, which we basically derived it from our massive data sets, empirically looking at the common microbes that nearly everyone has, and got a list of those that correlated with nutrition. So they were correlated with your food intakes, good and bad, either went up or down. They also correlated with health outcomes, good and bad. So those that were correlated both the food and the health outcomes, we then ranked them and got the 50 best and the 50 worst. And we've shown that's a much better predictor of health than the diversity score, and it performs better in our randomized trials, our longitudinal studies, etc. So it also works better when you're trying to compare vegans, vegetarians, and meat eaters who did that all got messed up with diversity scores, because often vegans might have lower diversity because they weren't having meat and dairy, and yet they were probably much healthier. So that paper should be coming out soon, and hopefully will set a new standard really for the microbiome. And it may evolve as we get as we work out we discover more microbes that might have even stronger effects on these. So I'm not saying it's fixed in stone, but the idea of ranking them without because everyone now focuses on a few of these microbes they know about, but most of the ones we found were unknowns.
Yeah.
They hadn't been discovered before, and suddenly they're having much stronger signals than these sort of household names.
Yeah. Well, Tim, thank you for all of the amazing work that you're doing. Congratulations to you and the team, and thanks for sharing it all with us today. If people want to follow you, follow your work, you're very active on social media, which I think is great. You actually take this science and bring it to people and help them understand it. Where's the best place for people to keep track of you and Zoe if they want to learn more?
Um, well, strangely, Instagram, which isn't most scientists go to, but
That's right.
I seem to be much more popular on Instagram than I am on Twitter these days. And Zoe's also on Instagram. That's more learning gut-friendly recipes and things. We also have a science nutrition podcast, which is very focused on nutrition, which is free, called Zoe Science and Nutrition. And Obviously, I've got books, popular science books. Got a cookbook coming out in the US
Oh, cool.
in about 2 weeks' time, which you get to practice all that science into 100 delicious recipes. And we've also got a free app coming out in which actually you can download now. You just go to it's a free app. Go to the App Store and Zoe Health app, and we're beta testing it, and it allows you to scan any food to see how risky the
Cool.
processing is, and you can actually photo log a plate of your food, and it will tell you how healthy you are. So yeah, there's lots of things happening, particularly if you're in the US, and you can also buy we have a product which is
I
called the
see it on the shelves in
the daily yeah
lots of
it's in
places.
called the Daily 30, and it's a prebiotic freeze-dried plants, and we did an RCT and it improved 40 microbes out of the top 100
Oh,
changed when you take that within a few weeks. So yeah, lots of exciting ways to improve your gut health.
amazing.
Most of them for free.
Yeah, tremendous. Well, thank you. Congratulations. And we'll look forward to maybe having you on in the future again as we start to scale these projects up, bring some of these new scores to people and see how it evolves. I'm really interested in following and diving in a little bit deeper. I learned a ton today that I just don't know about the field. So thank you for sharing it with us. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah, who produce the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.